So lactase gene that we were talking about, it's on chromosome 2. Now, alleles are kind of like, well, I guess we can use this analogy, kind of like basketball players. As you know, there's a difference normally between a shooting guard and a center. Or a point guard and a shooting guard. Okay, those are like different versions of alleles. Because of trade-offs. Usually you don't have an allele that makes you great at everything. There's always a trade-off. Okay, whether it's a trade-off in terms of metabolizing food in the old environments where food was scarce at times, people who had alleles that helped them survive in a food-rich environment, they got all sorts of metabolic problems that we find nowadays. Okay, so in the like the lactase gene, TAG. So this is peptides, the base pairs, and what happened was upstream there was a, let's see, is there a difference somewhere here? Oh yeah, right. So we can actually see right here. Okay, so good. Okay. Okay, so here we have, remember, you need 3 peptides for an amino acid. So GTG, and then it's opposite. You always have a G and a C, a T and an A, G and a C. Well, if you actually look right here, you've got CCC instead of GTG. Okay, and so this is actually upstream, and it keeps the lactase flowing. So it's a regulatory gene. Okay, you can understand this. Anything else? Okay, yeah. Not too hard. Now, it turns out that we can also use alleles to identify individuals. And in fact, what we do is we use what are called short tandem repeats. There's usually flanking regions around a gene that are repeats of certain lengths. And there's about 13 that we use. And it's highly unlikely that an individual who's not related to someone else they might share one repeat at one locus, but not a whole series of others at 13 others, right? And so it's kind of like a DNA fingerprint. And so we're going to be talking about short tandem repeats in a little bit. Short tandem repeats, they're found throughout the genome. It's thought that they don't really have much of a function, but maybe not. They might actually have regulatory function in terms of how fast reactions happen in terms of DNA transcription and translation. These are the chromosomes that humans have. We have 22 somatic chromosomes, not in our sex cells, and then we have a sex chromosome, an X and a Y. Females are XX, males are XY. So you can actually see these are the ones that are highlighted here. That's one of the repeats. That's one of the repeats. That's another one. 2 here. If you actually count them up, I think there's 13 plus the sex chromosomes. So these are considered non-coding regions. In other words, they don't code for a protein, but they still might be involved in gene regulation. But they tend to change a lot because they're not in a really strong selection. And so that's how you can actually, you see so much variation, you get to distinguish between individuals. So here's a repeat, a TAGC, and it's a tetra repeat. And you might have, let's say, so many of them in this case. You got 1, 2, 3, 4, 5, 6, 7, 8. Okay, at the repeat. Someone else might only have 6 or 5. Okay. We'll go through this in a bit, but we can actually measure how long repeats are. Everyone inherits 2 copies of each STR, one from mom and one from dad, just like an allele. We're diploid. We have an allele from mom and an allele from dad in the same gene. It may be the same allele, or it could be different. Okay, and so we'll get to these. Now that's good enough for now.